So every day the kidneys filter about 180 liters of plasma, concentrating it down to about 1.5 liters of urine that gets excreted from the body, taking waste products with it. In this lecture and the next, I'm going to talk about how they perform that function. So at rest, the kidneys receive about 20 to 25% of the body's blood supply. As it passes through the kidneys, the plasma is going to get filtered. And remember, plasma is the blood minus the blood cells. So as the plasma passes through the kidneys, it gets filtered out. And filtrate is the plasma minus proteins. So that's blood basically minus your cells and the proteins. Everything else gets filtered and becomes what's known as filtrate. That filtrate is going to get concentrated. Most of it gets reabsorbed. Most of the fluids and the ions and other solutes that are in the filtrate are going to get reabsorbed, so that very little of the original filtrate is going to be excreted. There are 3 processes that occur to form urine. It all begins in the renal corpuscle with filtration. The other 2 processes, reabsorption and secretion, occur only in the tubules. So the whole thing begins with filtration in the glomerulus, where we get rid of the cells and the proteins and we form the filtrate. Then that filtrate, we're going to reabsorb or reclaim much of the fluids, the water, and the substances, the solutes, like glucose and ions and whatnot, that are in the filtrate. And then we can add things to the filtrate as we concentrate it to form the urine, and that's secretion. So these terms are important to understand. Filtrate is the plasma minus the proteins, and the filtrate is what's inside the tubules. Reabsorption means that we're taking stuff back out of the filtrate. That could be water or solutes. And then secretion means we're putting stuff back into the filtrate. In the first process, we form the filtrate from the plasma that's circulating through the glomerulus. That filtrate is then going to pass into the tubules where the reabsorption and secretion occurs. Reabsorption, again, we take stuff out of the filtrate and reabsorb it back into, back into the blood supply. Secretion, we take stuff out of the blood and put it into the filtrate. The whole process begins with filtration in the glomerulus, where we squeeze the plasma out of the capillary beds there. There's water pressure that pushes the plasma through the so-called filtration membrane. No reabsorption occurs here, just filtration in the renal corpuscle. The filtration membrane consists of several layers of tissue in the renal corpuscle. It consists of both features of the glomerulus itself as well as the renal capsule that surrounds the glomerulus. And that filtration membrane is going to allow water and solutes to pass through, but not cells or proteins. So it begins with the fenestration of the endothelium of the capillary bed. There's a basement membrane in between the endothelium and the renal capsule, the podocytes. And those podocytes form filtration slits. So our incoming blood supply goes through the afferent arteriole, gets squeezed through this knot of capillaries of the glomerulus inside the renal capsule. This whole thing is the renal corpuscle. And then the stuff that doesn't get filtered continues through via the efferent arteriole. So the filtration membrane is found where this beige stuff, these podocytes from the renal capsule, converge with the endothelium of the glomerulus. And that's the filtration membrane. And here we zoom in a little bit and you can see the fenestrations that are a feature of the capillaries here in the glomerulus. These Swiss cheese-like pores in the endothelium there. And then the podocytes with the foot processes that come off from them. And again, this stuff, these cells are extensions of the renal capsule. And if we look at, like, say, a schematic of a cross-section through the filtration membrane, inside the glomerulus, the capillaries, here's where you will find the blood passing through. Fenestrations are these pores here between the endothelial cells of the glomerulus. So that's a special feature of this capillary bed. Then there's the basement membrane, which is just a little bit of connective tissue in between the endothelium and the podocytes. And then you have the filtration slits that are formed in between the processes of the podocyte cells. So these passageways for substances are too small for cells, much too small for cells, and too small for proteins. All other stuff, water, salts, glucose, etc., can pass on through here. The rate at which the plasma is being squeezed through the filtration membrane is known as the glomerular filtration rate, or GFR. And under normal conditions, about 25% of the plasma that's passing through the glomerulus is going to get squeezed through the filtration membrane, or that is to say, it becomes filtrate. So this GFR, glomerular filtration rate, depends on, in addition to the permeability of the membrane, depends on the pressure at which the blood comes through there. Much of the pressure is due to the water pressure. Again, that knotted structure of the glomerulus there squeezes the blood through at greater pressure. And the glomerular cells can change their size somewhat, which alters the surface area available for filtration. The more filter you have, obviously, the greater the filtration rate that it can occur. The glomerular filtration rate is carefully regulated by the kidneys for a couple of very important reasons. For one thing, the rate has to be high enough to allow the kidneys to efficiently filter out waste products from the blood. So the kidneys themselves have their own intrinsic controls. Intrinsic controls mean that this is from the kidney. So the kidneys maintain their GFR themselves so that they can perform their function efficiently. And the second reason that it's so important to maintain a constant GFR is because the body's own blood pressure depends greatly upon the function of the kidneys. If the GFR increases, the kidneys are producing more filtrate, which means they're going to produce more urine ultimately. If we produce more urine, we're going to lower the blood volume, because that's where the urine is coming from. So increase in urine means a decrease in blood volume. If the heart has less blood volume to pump against, it's going to have a lower blood pressure. And the same thing happens if we decrease the GFR, we decrease our urine output so that we increase the blood volume and therefore increase blood pressure. So there are these extrinsic controls, that is, they're external controls. They're not from the kidneys, rather the nervous system and hormones that maintain systemic blood pressure by affecting GFR. Of the approximately 180 liters of filtrate that we form each day with our kidneys, we only excrete about 1.5 liters in the form of urine. So the rest of that is getting reabsorbed. We're reclaiming the water and the solutes, returning them to the blood. This is known as reabsorption. Both active and passive processes are involved in reabsorption. And again, reabsorption occurs in the tubules, not in the corpuscle. There's a couple different routes that substances can take to get reabsorbed. In the transcellular route, substances are going to enter through the apical side of the tubular cells. So the apical side is that is on the interior of the tubule, so it's up against the filtrate. So that's where things are going to enter. They're going to travel through the cytoplasm, they're going to exit out the sides or the bottom of the cell, and enter the blood through the capillaries that are around the tubules. And then in the pericellular route, which is going to be preferred by water and most of the ions we would be concerned with, the pericellular route, substances are going to go between the tubule cells. And in most endothelial tissues, tight junctions that seal those cells to each other would limit the pericellular route. But in the proximal convoluted tubules, those tight junctions are a little bit looser, which is going to allow for reabsorption of these substances via the pericellular route. So reabsorb stuff out of the filtrate and back into the bloodstream. You go through one of two routes. Transcellular route, just come in the apical side, travel out either the basal or the lateral side of the cell, and make your way into peritubular capillaries. And in the pericellular route, you just slip through these leaky, loose, tight junctions, going in between the endothelial cells, and this occurs mainly in the proximal convoluted tubules. Sodium is the most abundant positively charged ion that you can find in the filtrate, and of the energy that the kidneys are going to expend on active transport, about 80% of that goes towards transporting sodium, and it gets transported or reabsorbed via the transcellular route. So it's going through the apical membrane and then the basolateral membrane. On the basolateral side, there's active transport. Remember the sodium-potassium pump, also known as the sodium-potassium ATPase. It's an energy-driven process. It's active transport. It's moving sodium out of the cell against its concentration gradient from low to high. So that requires ATP. Now, on the apical membrane side of those tubule cells, sodium is going to move via what's known as secondary active transport. So it's basically this diffusion here. Sodium is moving from high concentration in the filtrate to an area of low concentration in the tubular cells. And the reason that gradient exists is because of the ATP-dependent pumping of sodium out on the basolateral side. So because there was ATP involved in the first place, it's known as secondary active transport. And a lot of substances are going to be reabsorbed via secondary active transport because of the activity of the sodium-potassium pump. So virtually all of the glucose, amino acids, and things of that nature are going to get reabsorbed out of the filtrate. Almost none of that in a healthy kidney would make its way into the urine. And that's all because of secondary active transport dependent on that sodium-potassium pump. So there would be a carrier protein on the apical membrane side that's moving sodium down its concentration gradient. At the same time, it co-transports the other substance, the glucose, the amino acids, etc. The active movement of sodium out of the filtrate is going to create a gradient for water to follow via osmosis. Osmosis is going to be facilitated in the tubules by some protein pores called aquaporins, which basically speed up the process of osmosis by allowing a passageway for water out of the cells. So there are places in the tubules, proximal convoluted tubules, where those aquaporins are always in place, so water just flows through osmosis. Then on the other end of the tubules, the collecting ducts, the kidneys can determine how many aquaporins they need to have in the membranes there. And that's under the control of antidiuretic hormone. And we'll talk more about that in a little bit. As the water gets reabsorbed out of the tubules, it's going to increase the solute concentration of the filtrate. So now the filtrate is going to have a higher solute concentration than the concentration of the solutes in the capillaries around the tubules. So in other words, there can be diffusion now of solutes from high concentration to low concentration. And that concentration gradient, again, was established because water followed sodium through osmosis. So we pump sodium out to reabsorb the sodium ion. Water gets reabsorbed via osmosis following the sodium, and then other solutes get reabsorbed via their concentration gradients. And it all starts with the ATP-dependent pumping of sodium across the basolateral membrane, so the tubules. By doing that, we created a concentration gradient where we pump sodium out of the cell, so it's low concentration in here, high concentration in the filtrate. So sodium is going to diffuse. There's a carrier protein that facilitates the diffusion of sodium from high to low concentration. That carrier protein can be used to co-transport other substances down their concentration gradients. And the reason glucose would go from high to low concentration is because the water is leaving the filtrate, making the solute concentration higher here. And why is water leaving the filtrate? It's doing so via osmosis through aquaporins. It's following the sodium that was pumped into the capillaries. Most of the reabsorption is occurring in the proximal convoluted tubules. These tubules are in the cortex, and they're just after the renal corpuscle where the filtrate was formed. So almost immediately after we form the filtrate, we begin to reabsorb the really important stuff, our nutrients. much of the ions that we need, and most of the water gets reabsorbed almost immediately. The next part of the tubule after the proximal convoluted would be the nephron loop, which descends down into the medulla. There's different permeabilities on the descending and ascending limbs. So the descending limb is permeable to water, but not to solutes. The ascending limb is the opposite, impermeable to water, but is permeable to solutes. Now, even though the filtrate is going to go from the proximal convoluted into the descending limb first and then back up through the ascending limb into the cortex, think about what's happening in the ascending limb first. All right, salts can leave the ascending limb, water can't. So in the medulla we create a gradient, a salt gradient in the medulla, that water from the descending limb is going to follow. After the ascending limb of the nephron loop comes the distal convoluted tubule and then the collecting duct. And reabsorption here is under fine-tooth control. It can change depending on the body's needs. It's under hormonal control. So the collecting duct, antidiuretic hormone, influences water reabsorption by controlling the number of aquaporins. Remember, ADH is made in the hypothalamus, it's stored in the posterior pituitary, released from the posterior pituitary. It's going to act on the collecting ducts to increase the number of aquaporins. So a diuretic is a substance that makes us form more urine, like alcohol or coffee or soft drinks are diuretics. So antidiuretic decreases the amount of urine production. In other words, it has to increase water reabsorption. So antidiuretic hormone causes production of more aquaporins in the collecting duct membranes. It's going to lead to more water reabsorption, which means it's going to decrease the volume of the urine. Aldosterone is the body's main example of a mineral corticoid, which, if you recall, are made in the adrenal gland, specifically the zona glomerulosa. And mineral corticoids affect the body's ion concentration. So aldosterone acts on the kidneys at the distal convoluted tubule and the collecting ducts to affect sodium reabsorption. It does so by increasing the number of sodium-potassium pumps on the basolateral membranes of the tubules, as well as sodium channels on the apical membrane side. So increased aldosterone activity is going to increase sodium reabsorption out of the filtrate. Therefore, reduces the amount of sodium that we lose in the urine. And water is going to follow sodium, so aldosterone also increases blood pressure. ANP, or atrial natriuretic peptide, which comes from the atria of the heart, is going to have the opposite effect as aldosterone does. That is to say, it is going to decrease reabsorption of sodium. So it's going to increase the sodium that we lose in the urine. And that means it's going to decrease blood volume. So when blood pressure is elevated, the heart releases ANP. It's going to reduce the blood sodium levels, which means it also reduces the blood volume levels, because if we excrete sodium in the urine, then water will follow, which reduces blood volume, and that means that blood pressure reduces. And also, if you recall, parathyroid hormone, which comes from the parathyroid glands on the back of the thyroid gland. That's the body's main way of adjusting blood calcium levels, and it's going to act at the DCT, distal convoluted tubule, to increase blood calcium reabsorption, or I should say calcium reabsorption back into the blood. This figure summarizes the major reabsorptive processes that are going on in the tubules. So again, the filtrate forms here in the renal corpuscle, and reabsorption begins in the proximal convoluted tubule. There's no reabsorption here in the corpuscle because of the high pressure that the filtrate is being formed under. Here in the proximal convoluted, most of the reabsorption occurs. So we're taking back all the really important stuff that we need. So we don't excrete, we don't lose any of this in the urine. Also, we're reabsorbing much of the water and ions that we need. And then you have the nephron loop, which descends into the medulla. So in the ascending nephron loop, it's impermeable to water because this thick endothelial tissue here doesn't have aquaporins in it. So ions, however, are permeable in the ascending, and so the ions are reabsorbed into the tissue here and eventually into the capillaries, which would be around the tubules. So because we lose sodium here, we can lose water and do lose water over here. That is to say, it gets reabsorbed over here in the descending nephron loop. Then back into the cortex with the distal convoluted and the collecting duct, where the fine-tuned reabsorption of sodium and calcium is maintained. All right, again, in the proximal convoluted, just after we form the filtrate from the renal corpuscle, we're going to have reabsorption of most of the sodium ions, most of the water, all of the nutrients, glucose, amino acids, etc., in a healthy kidney are going to be reabsorbed here in the proximal convoluted. Many ions besides sodium, like chloride and potassium, bicarbonate ion, get reabsorbed in the proximal convoluted. Then next is the nephron loop. Remember, there's different permeabilities for the descending versus the ascending limbs. In the descending limb, you have permeability to water, but not to ions. And then in the ascending, it's impermeable to water, but permeable to the ions. So water gets reabsorbed via osmosis because of the ions that are getting reabsorbed in the ascending limb. They set up a concentration gradient here in the capillaries surrounding the tubules here, which water is going to follow. And then in the distal convoluted tubule, get reabsorption of sodium, and this is under control of aldosterone here. Also calcium under the control of parathyroid hormone. And then in the collecting duct, there's further reabsorption of sodium, also under the control of aldosterone, and there's reabsorption of water from the collecting duct to further concentrate the urine, and that's under the control of ADH, antidiuretic hormone, and it controls the number of aquaporins. Now the third process that's going on in the kidneys, in addition to filtration and reabsorption, is secretion. And secretion, like reabsorption, occurs in the tubules. Some secretion is going to happen in the collecting duct, but most of it goes on in the proximal convoluted. And secretion is basically reabsorption in reverse. So we're taking stuff from the peritubular capillaries, in the case of the PCT, taking things from the peritubular capillaries and moving them back into the filtrate. Some of the substances that get secreted would be ions like potassium, hydrogen ion, the ammonium ion, some acids and bases, the bicarbonate ion, and some drugs and drug metabolites, things of that nature. So some of these things would have been reabsorbed from the filtrate and then secreted back into the filtrate. But it's the kidney's way of fine-tuning the chemical content of the urine that's being formed. And this figure shows the substances that are being reabsorbed and where in blue, and the secretion in green. So in the proximal convoluted, you've got a lot of secretion of things like ions, hydrogen ion, ammonium ion, drug metabolites, etc. And then you've got some fine-tuning of the potassium content of the blood. Under the control of aldosterone here in the distal and the collecting ducts. And we also have secretion of urea here in the nephron loop, which is something I haven't talked about, but I will be talking about urea and its important role in maintaining this osmotic balance in the following PowerPoint lecture.